Okay, do we have most of the people from the outside? Okay, we're going to get started then. I'm going to have my two panelists introduce themselves. Kati, why don't you start? Say who you are, where you're from.
I'm Kati Juuti-Uusitalo. I'm from Tampere University, Finland, Europe. I'm a cell biologist. I'm creating very small intestines on the dish from the induced pluripotent stem cells, so that there are adult cells which are then introduced back to pluripotent cells, so that they can form whichever cell of the body except the germline cells, no egg and sperm. All the other cells of the body they can form.
Thank you.
So I'm Arnold Han. As I briefly said before, I'm a physician scientist. So I first became interested in gastroenterology when I was a medical student, and I decided I wanted to train in gastroenterology. I have a PhD back when I did my MD PhD. My PhD was in B-cell immunology. And when I became a postdoc, I became very interested in T cells. And so this melded perfectly. I'm one of the privileged, physician scientists that actually gets to see the very patients that hopefully I will affect through my research. So I see patients about 20% of my time, including celiac patients, and the other 80% of my time is spent in running a research lab.
So both of you are looking at inflammation, and in different ways, but very similarly as well. So the first question is, what can you tell the audience about the inflammatory cells that you're finding that may be inducing, or I would say maybe promoting, celiac disease?
Yeah, so in this sense, as I was talking to somebody before, we know a lot about celiac disease in terms of the inflammation. Okay, so we know that the first cell, the critical cell appears to be a CD4 positive T cell, which sees gliadin peptides, but only when they're presented by HLA-DQ2 or DQ8. So those are the genes that present gliadin peptides to the T cells. So the critical cell is a CD4 T cell, which is likely the driver and the main thing, absolutely necessary to cause celiac disease. And what I think is happening is that celiac disease is somehow co-opting our natural immune responses, which obviously aren't there to cause disease, right? So they're probably there to help us fight infections, and somehow your body, if you have celiac disease, is somehow misperceiving that gluten as a threat. So those CD4 T cells are key, but a lot of people, and what we're trying to understand is how that other cells besides the CD4 T cell actually coordinate to mediate disease. So we know now that in addition to CD4 T cells, we know that the IELs are the ones that are actually the end effectors that are killing the intestinal cells. And we have now more clues as a result of our studies on how those cells might be activated by gluten. But we still have a long way to go in trying to figure that out.
Kati, do you have anything to add?
Well, in our system, we do not have the inflammatory cells, unlike the organoids which are derived from the patient biopsies, which are the adult intestinal stem cells. But what we can look is what happens when the epithelium encounters with gliadin and with microbiome. And then, because we don't know what is the role of epithelium in this process, so that what we have just learned recently by Elena Verdu's laboratory, that also the epithelial cells are able to present the gliadin on their surface, so that they can be also the ones who trigger those CD4 positive cells to run the inflammation. And what we've also learned just recently, that because the gliadin needs to be deamidated by transglucosamidase to fit to the groove, which is then presenting those the gliadin to the T cells. So epithelium can also secrete the transglutaminase into the gut lumen, and so that the previously it was always thought that the deamidation occurs only beneath the epithelium. But nowadays it's also acknowledged that it can already happen within the gut lumen. So it has all the machinery to move forward with the actual inflammation.
Okay, well, that's very interesting because we know that there is treatment potential that could actually target that process. Do you think that that's something, I mean, I was a bit skeptical on it, but do you think that's actually something that may be helpful?
Sure, I believe that it is helpful. One of my colleagues from the lab next by, and also the pre-docs and post-docs of the Keijo Viiris laboratory, they are actually working in the same cell culture lab. So that yes, I do. I don't say that I do believe. I've actually read the publication, and then I do believe that it's because the recent publication by Valeria Dotsenko and others, if you're going to go and check, they've shown that the patient biopsies derived from the which have had the gluten, they are then taken and cultured in three-dimensional model. When there is gliadin, and then the ZED 1227, and then the inflammatory response with the ZED 1227, which is the transglutaminase inhibitor, it reduces the inflammation. So on a dish, it seems to perform extremely well. The best that so far has been shown in any other products.
Well, that's wonderful news. So that's good. What is your thoughts on even a healed intestine from someone with celiac disease, the still ability of it to be really diseased, I guess you could say. So I don't know.
I guess I can answer that question to some degree. Yeah, this is a question, right, where I think that there's a lot of other parameters of disease besides just intestinal damage, which is just one of them. So I think this is an important question that I don't think we've answered yet. For instance, there's data out there that maybe the antibodies have some sort of effect in and of themselves that are harmful, even if there is no damage to the intestine. So, for instance, that would be the case of potential celiac disease. I think there was a study recently that suggested that as a possibility. Or there's another possibility that maybe, that the immune activation, even if you're not getting disease, is that harmful in and of itself. So, I think that's something to be investigated, and I think that we can look at celiac disease as, a process that affects your ability to absorb nutrients, obviously, but also what other effects are there from that immune response. And these are things I think we're getting a better understanding of as we do more research in this.
Kati, yes. Chris.
Sorry, I'm citing again to the Keijo Viiris publications.
That's okay.
So, Valeria Dotsenko's and Keijo Viiris, the publication, I think it was 2019 or so. But then they what they did, they got the patient biopsies from the long-time gluten-free diet patients, and they assessed their transglutaminase levels, and then they knew that because Keijo is also a biologist, so that they got the samples, they knew what is the serology, and then they also had the crypt villus height from the biopsy samples, and then the crypt villus height was okay. And when they did also the transcriptome, so that they assessed what is the gene expression status, and then what they saw, that even those who had been in extensive periods of time on strict gluten-free diet, they still have a bigger proportion of those not so mature cells, which are coming from the crypt, and then they had bigger proportion of those, and then they had a smaller proportion of the absorptive enterocytes, which are the fully mature ones. So that there is something still going on. Of course, it can be that because there is always the inadverted the gluten exposure. So for sure, we cannot be positive that they haven't seen any gluten or gliadin for 10 years. Probably they have, but then it also says that even though that the crypt-villus axis looks superb, there might be still something going on, and then that the transglutaminase antibodies are also low.
Okay, because we do know that there is a disconnect also between symptoms and pathology. Is that the hypothesis, is that there really is still maybe differences in the enterocytes and what's actually happening?
Yeah, I think symptoms are really tough to figure out, right? Because they're different in many people, and I pay attention when patients volunteer for gluten challenge, and I always ask them, so what did you feel? And I get a wide, some patients don't feel much, but the most common thing that people suggest is they feel some sort of brain fog. They just don't feel right. And I think that, if you were to biopsy that intestine after a short-term gluten challenge, you would find it really looks relatively normal. So you're not, when people inadvertently consume gluten, or a celiac patient, has one dose of gluten and they feel terrible, it's not from their intestines being damaged, right? It's not from villus blunting. It's likely a result of the immune response that you're triggering. So I think that, some of these symptoms may be related to your immune response, and some of the symptoms from chronic celiac disease obviously are related to your intestine, and they're not necessarily the same thing.
So this is a big question. How do you see your research changing the way celiac disease is either diagnosed or treated? That's a big question.
Well, I think that's the hope of what we're all doing, right? I think that, what we've tried to do is understand mechanisms of disease, and I think that that's how most basic scientists think, right? We would love to cure diseases, and that's ultimately the end goal. But I think that if you're in the lab, you're trying to understand. That's your first objective, is to understand how things work, with the hope that eventually that's going to lead to opportunities to treat or cure those diseases. And I think we've made a lot of leeway in that. So we've grown to understand so much more about the mechanisms of what cell types are doing what and how they're being activated, and there's still a long way to go. But with everything we discover, it makes the whole disease make more sense from the standpoint of rational therapeutic design. So we're not just, now I think that translational medicine has come so far. Just 20 years ago, I think most drugs were just discovered by accident. It wasn't not that long ago. And now we're able to look at a biological process and specifically intervene upon it in many instances. So I think that that's hopefully coming in celiac disease. Now with the diagnosis, absolutely. You've already heard about, different ways people thought to look at diagnosis, either by the stool, for example, or by a short-term gluten challenge or interleukin-2, things like that. So I think that this has all come directly as a result of the research that we have done and many others have done to just try to understand the disease, right? So I think that it's already certainly affected diagnosis to some, maybe not as fast as we wanted to, but it has, and certainly given us opportunities for new diagnostics as well.
Kati, any?
I fully agree with Arnold with the biomarkers, and then those detection methods. But then I would also like to add that I hope to see that there would be also some kind of probiotic, so that you could have either to alleviate the reaction, or even to prevent the reaction occurring, either the probiotic or vaccine, so that if we were to understand the first step of the mechanism, then we would be able to step 2-3 steps backward.
So I actually also trained as an immunologist first. I have a PhD in immunology. And one thing that kind of frustrates me is that immunologists are not attracted to studying celiac disease, where it's such a perfect model. What is your feeling about that?
I think that's changing to a large degree. And I think another thing that's changed is just the interest of industry in general. Like we see, like a lot of, the pharmaceutical industry now is very, very interested in it. And it was not that long ago that people weren't. Now, I can only speculate on why that might be the case, but I think that, unlike a lot of other autoimmune diseases, you're competing with a pretty effective therapy, which is a gluten-free diet. So when we talk about immune diseases, some of the side effects from the medications are really, really terrible, right? A lot of immunosuppressive medications, when you suppress the immune system, the side effects are really, really serious. So people, for years, probably like, rather than give them immunosuppression, let's just do, a gluten-free diet. But I think that what has changed is obviously there's an increasing need, or there's always been a need, but people are appreciating now the really difficult to manage celiac patients, right, that don't respond or develop severe complications, even, no matter how hard they try to avoid gluten. And also, I think that we just gotten really smarter with our therapies, right? So as opposed to just hammering the immune system and just suppressing the whole thing, which obviously has a lot of effects, we've become more smarter about how we target the immune system and celiac disease. So I really think that that's changed. I've seen a lot more interest recently on the part of both academia and pharmaceutical companies in looking at the disease. So I'm hopeful about that.
Okay, Kati, anything?
I'm also very hopeful. So that there are two recent things that I think that are good to raise up. So that the IL-2 thing with the Jason Tidin and from Australia, so that you can induce the immune reaction. Even just by taking the blood sample and then adding the glidin there. And then also the Calvin Gur, the paper that the IL-7 is also there upwards from the whole signaling cascade. So those have been really, really fascinating results. And so usually when you have that kind of intriguing results, then that usually gathers more researchers to ponder around that question.
Let's be more hopeful then, I guess, that more people will be interested. I'd like to open it up for questions from the audience as well. If anybody has any questions, come up to the microphone. In the meantime, I want to know, Kati, you mentioned gut on a chip. Can you explain what that might be?
Well, gut on a chip is when there has been the theme, human on a chip. So at first it was a human on a chip, and so that you can have different cell types on a dish. So the dish is very miniaturized, like 2 centimeters times 2 centimeters, and then you can have the lungs, and then you can have the liver, and such things, like one after the others, and the cells are connected by little tunnels, so that the fluid flow is going through all those cell types, and then they are consuming the nutrients, but also also giving up, so that there is humoral connection. But also they're in the the human on a gut. So here the organ on a chip, gut on a chip. We could have it structured in that way that there is, in this case, the epithelial cells on top, and then underneath the porous structure there would be the endothelial cells, which are forming the blood vessels, and underneath that there could be, for example, the neuronal cells or some other cell types, so that you can have like miniaturized gut in there, and then usually those are automated in that way that you don't need to pipette anything, so that it's regulated, repeatable, reproducible, and then you can do the drug assessments or metabolic assessments from those, and then it's really, really high throughput. That is something that I see that those are the usage of those is increasing.
Any further comments about using this type of method or organoids and how that helps us?
Yeah, so that's something that we're very interested in. So just like this session is about different models of celiac disease, and I think that one thing I would emphasize is that the model we've used the most is just patients, right? And that there's so many different stages of disease in these different patients, and we've really leveraged that as much as we can, and I think that in and of itself has been invaluable. In most diseases, you only have active disease versus controls, right? In celiac disease, of course, you have gluten-free diet patients, you have potential celiac patients, and also you have patients who bravely volunteer to eat gluten, which has been also invaluable. And really, as an immunologist, this is a luxury you don't have in any other disease, right? You can't just shut the disease on and off just like that with gluten. So that is something that I don't think we've even exhausted yet. So we're really interested in pursuing that. Now, there are mouse models of celiac disease that people have developed that recapitulate many features of human disease, unfortunately, and we've learned a lot from them. But there are always differences, whether subtle or not, between mouse models and humans. So what we've done is we looked at organoids. So that's another way where we can, we've developed the technology in the past. I haven't developed it, but many people in the field of stem cell biology have developed these technologies where, normally you can't take a tissue out of a body and put it in tissue culture, and those cells will not stay alive, right? But people have figured out that if you keep the stem cells alive with the cocktail of factors, then you could actually take an intestine out and actually grow it for a little while. So, and there's a special technique of that where you can actually keep the immune cells alive in that tissue. So now we can do things like we can block interactions, or we can do more manipulation than we can do in human, obviously patients, right? So I think that's a frontier that we're working into and we're going into right now, where we can use that to specifically test mechanisms and block things and add things and see what happens. So that's been a really nice tool. I mean, Dr. Favell talked about a humanized mouse model, which also, it actually goes further than just regular mouse model, right, where you're actually using a human immune system. So I think there's a lot of exciting things on the horizon. And of course, the chip. The gut on a chip is something else, yeah.
That's great. Thank you so much for this panel, and I'm going to now hand it over to Lisa Fahey from CHOP, who is going to be the moderator of the fourth panel.